Tori is a dear friend and a wonderful scientist. Tori is the Diane Taylor Distinguished Professor in Neuroscience at Dartmouth College and the director of two centers, the Cognitive and Affective Neuroscience Center and the Brain Imaging Center. Professor Weger research centers on the neurophysiology mechanism of affective processes, including the pain, emotion, stress, and empathy, and how they are shaped by cognition and social influences. One focus area is the impact of thoughts and beliefs on learning, brain functioning, and brain-body communication. Another focus is the development of brain biomarkers, and if I understand well, it's part of their talk, and also a third focus that have all of us the development of statistical machine learning, computational advanced technique to help us to understand the complexity of our brain. Professor Weger conducts basic science in these focus areas and apply the resulting technique and model to collaboration around the world. We are very pleased to talk with us, and without any further ado, please.
Thank you, Luana. It's really nice to be here today, and just delighted to be able to be here and share these thoughts with you. So thanks for coming. Pain and pleasure are fundamental forces that shape our feelings, that shape our motivations, and that guide our trajectories through life, including changing how our brain learns. So as Jeremy Bentham said many years ago, nature has placed mankind under the governance of two sovereign masters, pain and pleasure. So it's really fundamentally important in all aspects. But when it comes to our clinical pain, pain crosses boundaries, doesn't respect boundaries across departments or disorders. It's a feature of many forms of pathology. And of course, the main reason that people seek medical attention in the first place. But when it comes to measures of pathology, we have very few measures of pathology. Sometimes we think we do. So for example, this picture here shows spinal stenosis that was diagnosed by 3 neuroradiologists. So clearly there's a cause for back pain there. Thing is, this patient doesn't feel any pain at all. So as it's now well established, MRI findings and pain levels when it comes to chronic back pain are dissociable. So as a result of the lack of biological markers for pathology, it's pretty difficult to treat pain often. So many widely used insurance reimbursable treatments, Medicare reimbursable treatments, are still widely used today, but they have been shown in meta-analyses to have no benefit over placebo, including arthroscopic knee surgery, vertebroplasty for osteoporotic fractures, epidural steroid injection for chronic back pain, of which my father has had 4. They last for 2 weeks and back. So we really don't have many treatments. And there's now a long litany of meta-analyses of pharmacological treatments for these common pain conditions like chronic low back pain, and nothing works very well. So maybe part of the problem is with how we're assessing pain. And of course, the gold standard measure, the only acceptable measure of pain, is pain itself. And this is the cup that was given to my wife when she was in the hospital with her 3rd child, and this is actionable. She could get different levels of analgesic or a medical intervention, or depending on whether she reports a 4, 4, 5, or 7 on that scale. The scale is highly reliable. It's a wonderful scale. It's easy to use. It's widely used. But there are problems, and the problems have to do with how complex a readout that self-report is. And to understand that, we can think about the case of getting a placebo treatment, getting nothing at all, except for the suggestions and information and context that comes along with being treated in various ways. Here's a simple laboratory test. We give people a 48-degree noxious heat on their arm, and we ask them to rate pain on a 1 to 10 scale. And we do this under 2 conditions. Under one condition, we tell them other people thought this was really low pain, not so bad. In another condition, we give them the identical heat, but we tell them other people thought this was quite painful. And we see a much larger effect. We've tested this in over 300 people now, And nearly every individual person shows the effect. So if this were a drug, this would be a blockbuster drug. The number needed to treat is one. But of course, none of us thinks that this is the solution to chronic pain here. We can also look at this through the lens of surgery. So many common surgeries, like for pain, obesity, gastric reflux disorders, and others, are widely used, and they weren't compared to sham surgeries. But later, when they are compared to sham surgery, which is making a superficial incision, not really doing the full surgery, the effects of the sham surgery are often as large or almost as large as the real thing. And as a case study, I'm going to go to one of the studies of arthroscopic knee surgery that was done by Bruce Mosley. So it was a sham surgery. And we're going to see in the video here where Dr. Mosley is talking a little bit about his motivation for the study, and you're going to see the setup of the study, and then you'll talk to a patient and his wife, we hear from them, who several years later can tell you about the pain before and the benefits due to being in a placebo condition, getting a sham surgery, even though he knows it's a placebo. So hopefully then the sound is going to come through.
We had a very elaborate ruse, if you will. I would play a videotape on the TV of a real surgery. And while I was watching the TV, I would ask for instruments and ask them back and forth in serial conversation as if it was what I would really have done if it was happening in the TV screen is actually going on in surgery.
Can you describe what pain was wrong before the operation?
I didn't say that it could be an influence. And yes, but maybe it could. Realize this. Clearly, cases we surgery, and as much before the patient is healing without the surgery that influences the result and the actual surgery itself.
All right, sorry for the echo there, that added effect, but you can see that this is a person who knows he received a sham surgery, but he doesn't care, he's better several years later. So when we report pain on scale, we're not assessing long-term function, and we're assessing something that's very complex. So when you report a 7 out of 10 on that scale, this is strongly influenced by your prior beliefs about pain, by, of course, nociception itself, but also by the emotions surrounding pain, by the reporting context. So pain reports are an interpersonal behavior or communication. And so we make communicative decisions. So it's not uncommon for people to report 11 out of 10 on a 10-point scale, or report a 10 because they're really asking for help. So then it's very hard to identify the pathophysiological component, let's say the nociceptive component, and treat that component. There's many other problems with introspection as well, but just one, people are really limited in their introspective capacities and their ability to report accurately what they're feeling at any given moment. And this is particularly true of vulnerable populations who have cognitive impairments, for example, or they're very old or they're very young, those mentioned. These effects are not limited to those cases. There are placebo effects compared to no treatment or treatment as usual in many clinical conditions, including multiple types of chronic pain: migraine, osteoarthritis, chronic low back pain, neuropathic pain, irritable bowel syndrome pain, labor pain. In depression, as standard clinical trial outcomes of 6 weeks, in Parkinson's disease and neurocorrelates of Parkinson's disease, in anxiety in emerging literature, in drug cravings and addiction, and even in some aspects and symptoms of schizophrenia. And finally, other bodily symptoms as well, including asthma. And we're going to look at a case study of asthma, because this is a particularly informative paper for us and a well-done one. So what Michael Wexler and Ted Kaczyk's group did in this study was give people in a crossover design albuterol, which is steroid inhalant, and the placebo and a sham acupuncture, and then a no intervention control. That's the baseline to compare the others to. And when it comes to subjective improvement, you can see people felt a lot better when they're getting all of these treatments, including a placebo and sham acupuncture. But they can also measure FEV1, or sex relation value 1, which is a primary behavioral measure of pathophysiology in asthma. And there, only the drug has an effect. There's no effect of the placebo or the sham acupuncture. So here there's a strong dissociation between symptoms and signs of pathophysiology. So we can look at this in two ways, two sides of the same coin. One is symptoms can be a fairly poor guide to assessing and treating pathophysiology. So we really need measures of pathophysiological mechanism, in this case, those that drive pain. But on the flip side, psychosocial context and care are really important for improving people's well-being, even sometimes over long time scales. These factors can complicate drug development, and what we really need to do is measure and control them alongside of pharmacological and other device treatments in order to isolate the effects of the treatment. So that's the first part of the talk, and there are 4 parts. The first part is about the power of placebo and difficulty that this poses with symptom self-reports, as important as they are. Next, we'll talk about the need for biomarkers, and I'll show you some results on fMRI-based biomarker for pain. And then I'm going to show you a little bit about how we can use those biomarkers to understand and compare treatments and understand which things have more fundamental effects on how the brain processes pain. And finally, we're going to go beyond a single biomarker and look at multiple biomarkers. There's not just one biomarker for pain, and use that to understand how placebo effects operate on systems related to fundamental deep sensory pain versus evaluative aspects of pain and avoidance. So what do we want out of a biomarker? Well, it's something that should respond to a known intervention like this noxious event. It ought to be a mediator of changes in subjective experience, in this case, pain reports, or any other outcome. And what we'd really like for it to be useful as a biomarker is these minimal criteria, not the only criteria, but it should be sensitive and specific. So it should respond whenever pain is present, and it shouldn't respond to other things that are not pain, or they're otherwise confusable with pain. And to the degree that we have those biomarkers, we can use them as targets for interventions. If we had such a thing for pain, then we could understand what are the effects of psychotherapy, what are the effects of gabapentinoids, what are the effects of neurostimulation-based therapies, and so forth, and how do those things converge. And there are many uses for biomarkers, but the one we often think of first is what's called a surrogate measure, a drop-in replacement for pain. And these biomarkers are not drop-in replacements for pain, and they shouldn't be. Instead, they can be used for many other kinds of uses. So they can be used as to aid diagnosis, to align the groupings that we give to patients and to individuals along the lines of what the pathophysiological mechanisms are and the brain processes are. We can use them as prognostic biomarkers to predict and track future disease progression, and then target those most in need of intervention with resources prior to something like surgery. We can use them as predictive biomarkers to help us predict who's going to respond to an intervention and who won't, and who might be harmed and who might be helped. And we can use them as pharmacodynamic or response biomarkers as a target for interventions that we can engage with treatments, multiple treatments, and then use to track outcomes. And as you'll see in the talk, I'll try to show you more about why I don't think that it should be a drop-in replacement for pain or a surrogate measure for pain. If you want to know how somebody feels, you ask them how you feel. But if you want to know why, what's driving that, or what's related to that in the brain, then you have the brain. Another benefit of fMRI or neuroimaging-based biomarkers in particular is that they can be linked to what one might call a nomothetic network, all kinds of different findings and aspects of behavior. So I can link MRI-based brain systems to PET measures of neuroinflammation and neurochemistry of various kinds, to animal system level analyses that get at mechanisms underlying pain and sensitization and so forth, to pharmacological findings, to peripheral inflammation and physiological findings, to our genetics and genomics in increasingly large samples these days, thousands of people, to our social networks, how we group and interact, and of course how we assign clinical diagnoses as well. So that's a rich way of understanding things. And I'm going to briefly critique what I call the blobs on brains approach, which is just saying, okay, here's a map, right? And let's look at all the areas that are activated during pain. So this is a real map from about 30 people. But what we'd like to know is, what does this map really mean? What is it telling us about a person's pain experience, or even what they're doing at all? And so often what happens is we see these blobs and we start to interpret them in light of what we already think, what our hypothesis already is. So we see that there's anterior cingulate activity, that's a receptive target. There's mid-insula and anterior insula and operculum, nociceptive targets, medial thalamus, another set of ascending nociceptive pathway targets, primary somatosensory cortex, and so forth. I go, oh, that must be pain. We could even do better. So Tal Yarkoni built this database a few years ago now, while using my lab, which has activation now. It's more than 14,000 cortex, all kinds of studies. And then you can plug this map into that and say, what do people talk about? What are the terms people use when they find results like these? And the top terms are noxious heat, sensory painful sensation, stimulation, muscle temperature. So think, aha, now I've got more confidence that this is really a pain map. The problem is, this isn't a map of pain experience. It's a map of what happens when you look at the person who rejected you in the above, which we'll come back to again soon. So in the old days, if you wanted to study pain systems or mechanisms, you could just deliver increasingly painful levels of a stimulus, see what lights up, and then look at those areas and say, does my intervention turn something down? And here, you find this is a mid-insula pain-sensitive region. It responds here. Its response to heat is reduced by an intervention. That's what you see in blue. And that intervention happens to be a placebo treatment. So maybe the placebo influences the pain processing, or how the brain is constructing pain. But if we look at even the most pain selective parts of this system, and look at those blobs, including the insula and the anterior cingulates, you can take the most pain selective part of the anterior cingulate. You can take that little bit of brain and ask, well, what are all the things that activate that particular area? And what's then the likelihood that it's pain given activity in that area? And the answer is, it certainly is activated by pain, but it's activated by a lot of other things. So the placebo effect on the anterior cingulate could be an effect on pain, but it could also be an effect on how we think about pain in a social context, or how we move, or the language that we use, or the emotions that we use to describe that. So this is a big problem. We really need better targets for those interventions. We need something more specific. So if we think about the literature of placebo effects on pain-related brain responses, this is large and growing. There's 100 probably studies at this point. We just did a meta-analysis of 600 people, and you see changes, right, in these areas. So reduced responses to painful events in the cingulate, and the insula, and thalamus, and other areas. Christian Buchel's lab has also found in a series of papers reduced responses in the spinal cord to noxious events with a placebo, and increases with a negative suggestion, nocebo. Activation of putative pain control systems in the PAG, the perioperative gray, and the rostroventral medulla. Release of endogenous opioids, which are typically pain relieving in the PAG and in the forebrain targets of sending projections, opioidergic projections. So all of this is really sort of wonderful, and evidence is accumulating. Many new studies, and many people here, for Luana's lab, for example, has contributed a tremendous amount to this literature. But we have to ask ourselves, are they pain representations, these things, even in the spinal cord? Is that a pain signal? And how do we know? And that gets pretty tricky to figure out. Are these activation of these systems, are they influences on pain representations? But that's the goal, is to get closer to that level of analysis, where we can be increasingly sure that the signals that we're seeing are actually reflective of the construct or processes like pain that we'd like to to measure. So what we really need is neuromarkers, which are predefined models, but it's not a moving target. I'm going to test the same thing again and again, so I can characterize that representation and see how sensitive it is, how specific it is. We want those to match how processes are encoded at the neural level, where there's many different populations of neurons that intermix. So we want to try to untangle those to the degree that we can. And we want it to be optimized to have good measurement properties. Sensitivity, specificity, reliability across retesting. It's a trait measure, replicability, reproducibility, and generalizability across cohorts and types of pain and other things that we don't think are that we think we should generalize over. Okay. We need to test this in new individuals and new cohorts to characterize this. So that's one of the major things that my lab has been doing over the past 10 years ago or so ago. We are developing models based on patterns of brain activity, some based on patterns of connectivity, some based on specific pathways that we are trying to match as closely as possible to what's happening in animal systems so we can develop closer correspondences, and sometimes dynamic models of how these processes evolve over time and are shaped by learning. And to the degree that we have provisional markers, then we can test their effects on interventions. And I should say that we have consistently preferred models that are as simple as possible and as interpretable as possible in order to be able to cross-correlate them with what's happening in animal models and in basic human studies and lesion studies. So I think that's really important. So this is not the big black box AI. I'm not going to talk about large language models here in this talk at all. So we really need to be able to interpret and make this match across species. So once you develop models and you characterize them, then it becomes really interesting to test what happens when you give somebody a placebo or an opioid or a provision of care by a supportive partner or caregiver who gets them and feel heard and understood. What happens with social support when you're having your hand held by somebody that's a support figure for you, attachment figure. What happens when you are engaging in mindfulness meditation and form. And we can, to some degree, put all these things on the same playing field. So this is the process and the first result for what we've called the neurologic pain signature, because that gives us a name to hang on and test across different studies. So that's the only purpose of this. And the way it was trained is to take a training sample. It doesn't have to be a big group necessarily. We can talk about that. But we take people and we take really good raters. We know that they're really good at judging pain. We've selected them essentially. And we know they're really reliable. And then we give them 4 levels of noxious input, from barely painful to as painful as they can stand. So we have this function in pain. And then we're going to train a model, which is a simple version of a machine learning model with some tips and tricks to stabilize the model and make it interpretable. And that's going to be what we're calling a signature. Not in the sense that it's a handwritten signature and it's unique in all the world, but it's simply a neuromarker, if you will, for pain. So we can look at this signature and we see that it involves activity in the anterior cingulate cortex and anterior mid insula, ventrolateral medial thalamus, S2 and posterior insula. So many of the targets that we can measure and manipulate in various ways in animals and humans. And we see that all of those targets are related to nociception. They're targeted by nociceptive pathways. But it's also that there's a particular pattern that we specify. So we'll test the exact same pattern over and over again with different samples and different characteristics. So it's not a moving target. And that's really important for reproducibility. So this is an example. We can apply this signature to new test data. We take people one at a time. We can apply the signature to any type of image. It could be the average over a certain number of trials of pain or some other condition that we think is confusable for it. And by applying the signature, I mean, we take a weighted average where the signature supplies the weights. And then we get a single number for that brain image, which is the predicted pain. Now we can treat that measure like any other measure and validate its sensitivity and specificity, et cetera, across different cases. And everything I'll show you here for the rest of the talk, these models will always be tested in new individuals so that they're unbiased. They're fair tests of how large those effects are. Okay, so this is an example from another study from a different scanner and different population. We have Each line is a person. These are 46, 47, 48 degrees noxious heat. And we can see the lines go up for just about everybody. So that's a basic validation of increasing with increased pain in a new sample. If we turn that continuous measure into a classifier, we can say if we want to guess based on the brain activity whether a person is showing high or low pain or feeling high or low pain, we can guess right about 96% of the time in this sample. And that's been a benchmark across different studies. And so then you might immediately be asking yourself, well, what is it actually measuring? There's a lot of things that go up linearly approximately when pain goes up. So it could be a specific subtype of pain. It could be heat pain on your left arm. That's what it's trained on. It could be some broader subset of somatic pain experiences. It could be anything that's negative affect, or maybe anything that's arousing or anything that's salient. All those things are going up at the same time with pain. So how do we tell? Well, I don't think that we can have pain and subtract all of the arousal and salience from it. If you know how, let me know. But what we can do is we can try to independently manipulate arousal, salience, negative affect in ways that are not pain. There are other types of intervention, and see if our markers are sensitive to those. Here's first data point on this. And this is from a large meta-analysis, which the placebo imaging community had pulled together into a community and we've pooled data across 600 people in 20 studies. It's now being expanded even more. So people are sharing their data, and we're testing this in more and more rigorous ways. And this is the sort of first result from that, which is, does the NPS actually respond to pain if you take different types of pain in different cohorts mainly from the US and Europe? And the answer is it does. So the shift to the right means that study or an individual shows a significant response in the right direction. It's more activated with pain than at rest. And the small dots are the individual participants. So what you can see here is across these diverse set of studies, not optimized for this purpose, the same pattern responds to noxious stimuli that are electrical, heat, laser, mechanical, and visceral, so multiple types. And it's going in the right direction in 95% of the people. And the effect size across this set, which is unbiased, is over 2, which is a massive effect size. It's close to the level which you would really need to detect things in individual people. And if you might know, I'd love to talk. I took out some slides here because time, but there's been this really interesting paper in Nature last year by Sean Merrick and Brendan Turbo-Clemens and colleagues, where they analyzed individual differences in brain activity, predicting all kinds of different measures: cognitive function, psychopathology, age, task performance, and so on. And the quick take home from that is that the effects are really tiny. They're really tiny. They're 0.1, or at the largest, something like, the largest even with multivariate prediction, 0.4, the largest effect size. So this is 10 times larger than that. What's different? Well, they're using resting state and brain structure to try to predict individual differences with tons of different compounds in large samples. We are targeting a brain state, the experience of pain, with a task that's designed to elicit that experience. So we could love to talk more about it, but I just wanted to benchmark this. So that tells us that it's generalizable and it's robust. So how can we manipulate things that are not pain? Well, we can turn to rejection, which is something that happens early in life and can often last a lifetime and be extremely aversive. And we had published an earlier paper in PNAS, which is the one that generated that map that I showed you, the rejection map, actually. And what that paper showed was that rejection evoked similar activity in many areas of the brain, including the anterior cingulate and the insula, and even the posterior insula, which is otherwise somatic specific, which is spawned many headlines and helpful points of advice, like it's really pain in the brain, that rejection is like pain in the brain, and it really hurts in how you respond to save your life. So, all right. So lots of sort of simple take homes from the media about this, and that lets us. They are very similar. They're very similar. So that lets us take a closer look. So in this study, Ethan Cross recruited 60 people who were all romantically rejected, they all brought in pictures of their ex-partners and pictures of a close friend. So for example, if one of our sample participants who might live in Hollywood might bring a picture of a friend and an ex-partner, this wasn't real one, and then they experience painful and non-painful heat in intermixed trials. So this is then allows us to both validate on pain and test the effects on rejection. They have similar negative affect ratings across the two, rejection and pain, similar brain activity in terms of which blobs are activated. So now we can apply the NPS and ask, does it discriminate hot versus warm if it's physical or somatic pain? And the answer is yes, we're about 90% accurate. And that's with about 8 trials per temperature per person. So not too much data per person. And then we can ask, what about the rejector? And for me, there's a few moments in science when you have this sort of aha moment where it could be interesting either way it turns out. You know, I thought maybe rejection really is like pain, maybe it's not. Let's see what happens. And we found that it wasn't. There's no significant response. And then you might think, well, we could test this lots of ways at the moment of rejection, at the moment of grief. What about people who felt pain? There's many things that we could do, but we tried pretty hard to find correlates of this. You know, it's not just the people who, it's not true for people who self-report that it feels like physical pain, for example. But so on average, not so much of a response. And here what you can see is the NPS expression of the response when it's actually pain in red, and then when it's non-painful warmth in orange, and then the rejector and the friend are in blue. The dashed line is the threshold that we use across studies to classify pain, no pain. Right? So it's not that the NPS never shows any modulation about anything that's not pain, but if it's above that line, we think it's pain. There's nothing that looks like that here. Okay, and then you could say, well, you know, what about the anterior cingulate? Because the anterior cingulate is the convergence zone, right, where there's pain affect. That's where it's really happening. And the anterior insula, that's a convergence zone. So we can look only within those areas. And this is the anterior cingulate as an example. And we can take only the voxels, bring this in this area, and we can retrain a model that can accurately classify whether it's high or low, painful or not, if it's somatic. But it doesn't care about whether it's a rejector or a friend. There's no signal. We can take the exact same set of voxels, and we can train a model that can detect rejector versus friend in the anterior cingulate cortex, but it's at chance for high versus low pain. So that pattern is called separomodifiability, which is Saul Sternberg's theoretical work, and that's the strongest criterion I know of for separability of processes. What it means is that we have 2 different interventions, each of which affects one and only one brain measure. And those brain measures are intermixed in the anterior cingulate, because the anterior cingulate has neurons that do lots of different things. So what this tells us is that we can unmix pain and rejection to some degree. And we can take it a step farther, because many times people are really interested in connectomics these days. And what you would normally do is take that area of anterior cingulate as a seed region, let's say, and you can correlate it with other things in the brain and analyze its network properties, and so on. But the claim here is that this is a mixture of multiple things that are different affective processes that we can disentangle. So how can we test that? Well, we can take the local pattern expression in the anterior cingulate, apply that to the time series of images, so we get for every person a pain-related anterior cingulate pattern. We can apply the rejection pattern and get a rejection-related anterior cingulate pattern. You can see that they're not correlated, and then we can correlate each of those with other areas of the brain and ask, where do they converge and diverge? And they diverge strongly everywhere. So the connections with the pain pattern include many of the typical targets that you would see for pain here in red and a lot of stuff. The connections for rejection include things like the ventrolateral prefrontal cortex, which I actually think is super important for multiple types of aversive and other feelings, generating feelings that are not pain related. So different targets we can disentangle them. Now, what about vicarious pain? If you see somebody like this, I feel something in my stomach, right? Is that like pain? Does it co-opt your pain system? Well, the theory has been from Tanya Singer's famous science paper that there's a shared basis for pain affect in the anterior cingulate and the anterior insula. So we can look at that, and we can do an even more elaborate manipulation where we have 3 levels of somatic pain on 2 sites, upper and lower arm. And we have 3 levels pre-calibrated of vicarious pain, watching pictures of other people in pain on upper and lower arm. And that lets us test the activation overlap, which there is. They both activate the same broad regions. We can then test pattern similarity in our markers, and we can also even look at somatotopy. And there's lots of results on these I'm not showing you, of course, for time reasons, but all of those things are in the papers. So for both, again, somatic pain and vicarious pain, people report similar levels of negative affect on a scale, and they increase across the levels you see here. And now we can, again, validate our NPS measure and say, does it respond? And the answer is yes, it does. It tracks pain intensity quite nicely and stiffness intensity across hand and foot. What about vicarious pain? Does it respond to watching other people in pain? And the answer is no. There's nothing about zero effect that's strongly deactivated overall. Okay, so there's transfer and there's no response to vicarious pain selectivity. Well, maybe there's just no signal to vicarious pain. Everyone's doing something different. Well, it turns out, this is our pain signature responses then, selected for pain. It turns out that you can actually decode a reliable signal that predicts how bad people feel when they look at other people in pain. We called it vicarious pain signature, and you can see that here. So it goes up across 2 body sites, independent, both body sites. When people are looking at other people in pain, it tracks their feelings. There's no response to experience pain at different instances. So that again is the separate modifiability process. We also found strongly divergent somatotopy, where you found expected somatotopy when it's actually pain. You see nothing like that somatotopy when it's looking at other people in pain. So there's somatotopy in different areas. Okay, they're different systems. Another controversy in imaging has been about reliability, where it turns out if you look at region by region, individual regions, the reliability is really bad, 0.4, 0.3, something like that. Pain ratings are really reliable. So here in this set of studies, each dot here is the study, so there's 9 studies, I believe. Pain ratings are really reliable, above 0.9. NPS responses, a test retest, are almost as reliable. And we think that there's actually long-term stability across at least a month, and we haven't tested it beyond that. And some of the other subregions here also have high reliability. In this study also, it's a different set of studies, but they're very large effect sizes, just like we've done before. And the interesting thing here, I'm just going to say it, I don't have a graph here, but what we found is that NPS values are reliable, pain reports are reliable, but those two are only moderately or weakly correlated. The correlation is much less than the reliability. Above 0 is weak. So what does that mean? That means that the NPS isn't recapitulating pain as a measure. It's actually, measuring something different. What's different? Well, think about what happens when people rate pain. I'm going to ask 2 of you to rate pain on the scale. After I pinch your arm or something, right? Are you really sure that you're using the scale in the exact same way? If you report a 7 and you report a 6, does that mean that you really feel more pain? No, right? It depends on how much pain you can imagine and what happened to you before and all these complex contextual factors. So one of the sources of divergence is all of the complex context things that we were talking about. And MRI has its own sources of artifacts that we need to try to contend with, and sources of noise that I'll go into later if you want to talk about it. So, after all of these tests of a decade or so, the take home here is that it's really tracking somatic pain across multiple types, especially for hypnosis-sensitive pain. Okay, so now we can use that to help us to understand treatments and so we can ask, which treatments have the largest effects on the NPS as a proxy for nociceptive pain systems. And we can test things like cognitive reappraisal, near and dear to my heart, asking people to think pain up and down in some blocks. Imagine it's burning, sizzling, bubbling. In other blocks, imagine it's spreading warm. This is like a quasi-hypnotic sort of suggestion, I'm told. And we can ask whether the effects are on the NPS, which means they probably have early effects on processing, or are the effects independent. So pain ratings go up when your reappraised pain is worse and down when your reappraised pain is better. How about the NPS? It tracks the stimulus intensity and pain quite well in this study. Again, no effects of self-regulation. We don't think it's very easy to think it up and down or imagine it up and down. Let's go back to our placebo consortium. So all of these 20 studies also had a placebo test intervention. So we can say how big are the placebo effects? And the placebo effects are really small. Across 600 people, they're barely significant. The effect size is 0.07. So it's tiny. They are correlated. Again, the correlation is weak, with the placebo analgesia that the person reports. So we think that this is not much of an effect. This is not the whole explanation for the much larger behavioral effects, which are about 6 times larger, actually, in this sample. The effects on pain reports. What about mindful acceptance? Something I'm deeply skeptical of personally, right? Depends on the kind of mindful acceptance that one practices. But yeah, what about this? Does this have an effect on pain? Well, it does have an effect on how people report. When they're reacting in red versus accepting in blue, there's a change in negative affect. It goes down when you're accepting. That's great. What about the NPS? Well, here, there actually is a significant drop. This is a small study. We went to a big one, but it's about a 25% drop in values. What's the difference? We don't know. But maybe adopting a state of mindful acceptance, adopting a stance and practicing that and getting yourself into it is different than sort of rethinking, reframing up and down, or getting a sort of standard placebo treatment. What about supportive touch? In this study, women came in and their partner came in and held their hand while they experienced pain, or they held the squeeze ball. And what we can see is NPS responds beautifully when it's supposed to during heat, not during other phases of the trial, anticipation, or report. And what happens to the NPS with supportive touch? It goes down. The squeeze ball in blue and the hand holding is in red. So there's a significant reduction there. Okay, so we think that some of these interventions work better than others. And maybe that can help us guide how the interventions are unfolding. However, leading to the last part of the talk here, there is no one pain signature. So this is a measure that's related to pain in some cases, some of the time. It doesn't track the pain influences of all these different manipulations. Some of them are going through different pathways. And if we turn back for a moment to chronic pain, there's this wonderful literature that's emerging and growing, showing that with various types of chronic pain are quite different in the brain in animals. And there are changes. You can look at neuroplastic changes. There are different changes for different types of pain in models. There are different interventions that can reverse or prevent chronic pain. So it's going to be very context dependent. Our job is to figure out what the dependencies are and where are the points of consistency. And I'll group these broadly into those that are sensitization of nociceptive systems, engagement and plasticity in pain avoidance systems that lead to hyperavoidance, and a shift from descending inhibition to descending facilitation are 3 of the different kinds of processes that we can say. Okay, we don't know that much. We're still learning a lot about how they apply to human pain. Now we have our NPS, and this is again back in the realm of evoked experimental pain that we can control tightly. And what we did is we took a sample of 180 people and we removed the NPS statistically, and we removed all the stimulus intensity effects statistically. And we wanted to know what predicts pain independent of all those things. So is there something, an endogenous contribution to pain? And there is, and this is what it looks like. It's very strongly related across multiple studies in this sample to pain. And there's again many features of it, but some that I'll point out are deactivation of the ventromedial PFC and a pattern of deactivation and hyperactivation in different parts of the nucleus accumbens zone that seem to match what we think might be going on in some animal studies. So then we can apply this pattern as an independent marker. And when we look at this idea of reframing or rethinking pain and ask what mediates it, it is in fact these regions, the ventromedial PFC and the accumbens, that as you reframe pain to be less aversive, you activate those regions. And the more you activate them, the more pain goes down. And no other areas in this study that we found. So now we've got 2 target markers. We've got our NPS, which we think tracks nociceptive pain. It's pretty hard to influence with placebos, and we think. And we have a stimulus intensity independent pain signature, which seems to respond to self-regulation. And it also mediates the effects of expectation and perceived control in some other studies in that original paper. So we can go back to the placebo case. And we need larger samples to really unpack these things and the individual differences. So now we're running a sample. We have about 500 people run. They're twins, so results are starting to come out. This is an early look. This is the first 400 or so people where we pre-registered some basic analyses about placebo effects. And there's a pretty standard placebo paradigm where people get a control cream and they get an identical cream that we tell them is an effective analgesic cream. It's lidocaine. It's going to block pain sensitivity. We apply that to different fingers, and we do a preconditioning or manipulation to convince them that it's really true. So we tell them that they're going to get stimulation at high intensity for both, but in fact, when they're being stimulated on the placebo-treated site, we reduce the temperature surreptitiously. Then we test different fingers to avoid a compound, and we're going to test them with heat. And we also have a transfer condition, which is pressure, which there's no training on and no conditioning. So we like to see if the placebo effects transfer to painful pressure. And all those are the same stimulus intensity. And this is done in the scanner, so it's kind of an elaborate protocol. So how does it work? Does it reduce pain reports? Yes, it does. So we can see sensitivity of both heat and pressure to the stimulus intensity level, which is very nice and important. We can see a fairly substantial placebo effect. That's the red is control and blue is placebo in both heat and in the pressure transfer condition that they didn't experience before. So those are the placebo effects. The effect sizes are very similar to the other meta-analysis, about 0.6. Now, what about the NPS? Well, it does track. It's sensitive to heat and pressure intensities. And what about the placebo? There's no placebo effect. There's no reduction as expected at this point, right? And even some of these areas that are involved, like posterior insula, for example, it's actually more activated with placebo. But I'll have to do a part 2 talk another time to unpack that more. So that we're really intrigued by that. What about the stimulus intensity independent pain signature? There we do see placebo reductions. Again, the signature response is sensitive to heat is reduced, and it's even reduced in the transfer condition. And interestingly, I think individual differences, I think, are going to be really tough to predict for brain activity, like that Merrick IASP paper that I told you about. But here, what we can see is with a large enough sample, we can feel really confident that there is a correlation between the analgesia and the SIPs. So the larger reduction in SIPs, the larger the analgesia. So, where does that leave us? Why is this? What does this all mean? Right? Well, we think what's happening is that we have multiple targets. We have nociceptive pain system targets, but in patients, we have sensitization over time, where there's a shift towards more importance of fear and avoidance pathways. And these are looking more like frontostriatal pathways. This is more like the SIPs pattern. And this is perhaps why, if you turn to clinical pain for a moment, we see positive correlations in multiple studies with spontaneous back pain in this frontostriatal system, prediction in some studies of the transition to chronic back pain, shifts in fibromyalgia that I'll mention, connectivity that increases with the insulin failed back surgery syndrome, connectivity with posterior insulin back pain. So these areas, which are often associated with the default mode network, are really showing up as important in chronic pain. If we look at fibromyalgia as a case study, what we can see here is that if we give people with fibromyalgia and controls the same level of noxious input, the patients are more responsive. So there's hypersensitivity. And that hypersensitivity mediates hypersensitivity of both pain and sample. But that's not the whole story. Most of the patients really don't show good evidence for that, in fact. And instead, there's other things that are much more strongly predictive of fibromyalgia. So if we put together this evoked hypersensitivity with another pattern trained on non-nociceptive stimuli at all, just visual checkerboards. I mean, people don't like them, right? The patients don't like them. They find them aversive. That's part of a classifier. They show altered brain responses, and they show alterations in these pain regulatory systems and their relationship with pain. If we put those things together, then we can classify patients versus controls with about 90% accuracy in a fair test. But the smallest of those effects is really the evoked hypersensitivity. The largest of these effects is this multisensory sensitivity. And it's the multisensory sensitivity that correlates with spontaneous fibromyalgia pain, not the evoked hypersensitivity. And I'll do one more brief case study here, looking at race and ethnicity differences, which is increasingly important to understand in terms of health disparities. So it turns out, particularly African Americans tend to report greater pain from the same stimulation in laboratory tests, and they have higher prevalence in many clinical pain conditions. The causes are unclear, but we did a brain imaging study to look at the NPS and what else might be happening. And what we find is that the African Americans in this sample versus white and non-white Hispanic are in fact hypersensitive. They report more pain in our experimental thermal pain tests, across temperatures. And this hypersensitivity correlates with measures of perceived racial discrimination in life. It doesn't correlate with, and our African Americans are not different in many other things like mood, socioeconomic status, catastrophizing, and other measures. So really the discrimination was fairly unique. And now we can say, what happens with the NPS? Well, we see very similar NPS responses across all of these different intensities. And we see similar correlations with pain reports across all the intensities in all of the groups. So we don't think that the NPS is different, but you can see the correlation with the pain reports is getting a little bit weaker. We think that's because there's something else going on that explains that hypersensitivity. When we look for mediators of hypersensitivity, what do we see? Measurement of prefrontal cortex and nucleus accumbens. That same frontostriatal axis. And particularly in our African American group, it starts responding more at high temperatures. It correlates more strongly with pain. There's a shift from anti-pain to pro-pain, just like our fibromyalgia patients. And this mediates effects on pain reports, and it correlates with measures of perceived discrimination. So we think what's happening is that our African American participants are coming in, they're not feeling very comfortable, they're not feeling very safe in this environment with a weird MRI machine. And they're, justifiably, their decision value of pain is different. They become hypersensitive, and it's being driven by this system, but not by ascending nociceptive systems. So to summarize this, then we have different pathways for different interventions. So things like mindful acceptance and supportive touch and conditioning, which I didn't show you, so I put a star by it in this talk, really seem to have these very early effects on nociceptive processing and the NPS. The effects of placebo and self-regulation and perceived threat value of pain and social information, like I showed you in the beginning, we tell people, other people think this is high or low. They are mediated by these frontostriatal systems, but not by changes in early sensory processing. So we have a association. So to summarize, I've tried to make the case here today that placebo treatments can powerfully affect symptom reports and clinical outcomes, but the symptoms themselves, as wonderful as that is, are a fairly poor guide to pathophysiology. So we need biomarkers, and we need biomarkers for the processes underlying pain to guide our understanding of the pathophysiology and what the effects of placebo and other interventions are on that. So imaging-based biomarkers can track pain with high sensitivity, specificity, and generalizability, at least in certain cases I showed you, but it's evoked pain, brief evoked pain. And that different psychological interventions have different effects on that system, and we can use those to help order them. So mindful state-based training is better than self-regulation and placebo treatments, at least as operationalized. But of course, the placebo universe is a universe of different things that you can do in different kinds of interventions. So we have much more to explore there. And finally, the typical placebo effects are those, I lost the whole delayed on the slide there, are those on pain evaluative systems that are involved in chronic pain and involved in health disparities, but that are dissociable from nociceptive systems. I'd like to thank my lab and graduate students and postdocs who are doing a lot of work. I'd like to thank our collaborators and formal lab members, of which there are many, and it's just very grateful that we are really forming a community. And I'd like to thank our Boulder Pain and Placebo genetics team, in particular for that last study. It's been a big effort. I'd like to thank you for your attention.
Thank you, Thor, for this wonderful talk. Thank you, everyone here and those on Zoom from IASP and other collaborators and members. So I think we have time for a few questions. Some are in the chat, but we give room for the people here in person to ask questions. Somebody.
Hi, I'm Amber Plattner. I am in the Department of Pain and Translational Symptom Science, and I work in the cancer population, which is kind of unique in that we can oftentimes predict when somebody might get pain, if they're going to have surgery or radiation therapy. Do you think with your neurologic pain signatures, if they're going to be accurate enough? I mean, so you said that of course patients are coming into these situations with huge different backgrounds, and they might not even have like the language to describe pain in the way that they might have to experience it. But like, do you think that these signatures are going to be accurate enough to be able to predict who is going to get pain or who might benefit from some of our interventions?
Yeah, so the question is about whether these kinds of signatures or these signatures are going to be sufficiently accurate to predict pain and who's going to respond to an intervention. I think that's one of the things that it's really, I think, important to test as a next step. The hope is that what it could do is, for a given population, a group of stratum of patients, is just to tell you whether their pain is likely driven by sort of nociceptive input, or they could be a good target for peripheral interventions, or is it really driven mainly by sort of call them threat and avoidance systems, right? And learned plasticity in those systems, frontostriatal systems, let's say. I think we have more to do, but I think there's a good chance that could be tried. I don't foresee everybody in the world, all patients getting brain imaging. I don't think that's going to be practical. But the hope then is that we can identify these classes of people, then better characterize the pathophysiology in those subgroups, better characterize behavioral measures that we can assess in those subgroups, and then use those as selection tools. But yeah.
Other questions? Yes, Andrea, and then we go to the chat. Andrea.
Okay. I was going to ask whether you ever get concerned or think about the majority of your data has been collected in a supine position, which is normally an escape from pain position. Usually when we walk around in the world, we experience pain either walking or sitting down, and how that might change, especially given your conversation about the context now. Lying down is almost immediately a rest or a safe position.
Yeah, that's right. Because the question is, are we worried about testing everybody in a supine position?
I don't know how you do it otherwise.
Yeah, right. Yeah, good question. Yeah. I mean, I do think about that. I even think about the fact that every time we do a pain challenge in the scanner, we're giving them something, and they've agreed, they know it's coming, they actually signed up for it. They're not, scared of it usually, right after the first experiences. But if they really are, they want to get out, right? And I think the lying down is actually potentially a surrender position. So if you go in for acupuncture, right, you lie on the table, you're in no position to really escape quickly, and you subject yourself to needles and things and hot rocks, on your body. So I think this is actually quite an interesting posture in terms of the acceptance. So we might be shifting people towards acceptance. In our healthy populations, normally ventral medial PFC that comes with activity is associated with less pain. It's pain off. And there could be a dynamic balance where in our patient groups or some of our American groups, we actually see a shift so that activity there becomes more pain on. So I think that the position itself could actually play into that dynamic balance. Is there a question, Jack? Because I put Q&A. Aha. Okay. So let's see. Any studies of people with chronic primary pain, IBS, fibromyalgia, TMD, etc. Yeah, the fibromyalgia one, we've looked at whiplash pain, we'd love to test a lot more things, frankly. I think part of the idea, all of these things are actually available on our website, all the signatures. So we'd like to test them. We'd like other people to test it too and see what happens. And we're working on some collaborations with functional pain syndromes, or and what we actually see is that the NPS actually seems to be responding to emotional events, to non-painful emotional events in a couple of different patient groups. So that's really interesting, which is sort of trying to make sense of that. And I think also includes our chronic back pain sample as well as fibromyalgia and IBS groups. So yeah, if you're interested, then try it out. I'll help you test it and apply it to your samples. I would love to see more of that. Okay, let's see. I think vicarious reporting of pain could apply to sport. Amateur marathon runners see other other running sports. Okay, I think I get it. Okay, so for example, if I'm running with it. Oh yeah, okay. I'm going to paraphrase it because I'm struggling with the wording of the question. But if I'm running a marathon and Milano's running next to me, which could happen later today, and I look over and Milano's looking like she's in a lot of pain, right? Then it's going to be contagious, right? Am I going to feel going to be in pain too? And I think that's likely to happen. I think that's actually pretty important. We did one study of placebo effects with 3 experiments that was done in New Chang's lab. What they did there is they influenced a care provider, simulated care provider, to believe that they were giving an effective treatment versus an ineffective one. And when they were giving an effective treatment, their pain facial expressions went down and the patient's pain went down too. So I think that's one example of interpersonal transmission. And I know Milano's working on other examples as well. Let's see, here's another one. Okay. How is the performance of the signatures in predicting nocebo hyperalgesia? Okay, which is the sort of flip side or twin phenomenon to placebo. The answer is we don't know. Let's test it. We've just been comparing placebo to control usually. So we haven't done a lot of nocebo studies, but I'd love to see how it works. Oh, I think Christian Buchan's lab actually did, with Stefan Goiter, did actually publish one in eLife, where they actually did use the NPS. And yeah, that's right. I'm trying to remember if it's causal hyperalgesia. I'll have to look back. So question, do you think the relative increased effective mindful state-based training is just more distracting than self-regulation? Oh yes, I like that question. We can look at distraction. It's hard to fully disentangle the effects of attention from these other effects. But what we can do is, and we did this in the hand-holding study actually, and I think the same will be true in the mindfulness study, where we can look at the brain networks that typically respond to distractors and cognitive demands, dual task demands. And what we can see is that the mediators of the hand-holding effect of these are very different from those distraction-related regions. So the distraction-related regions are frontoparietal, dorsal frontoparietal systems. The mediators are orbitofrontal, ventromedial PFC autonomous, those systems. So we think it's quite a different system, but we'd love to test it more.
First of all, wonderful talk. Thank you so much for the session. Two questions because I'm reading. First question, it sounds like the answer to this is yes, because you've been talking about fibromyalgia patients too. Have you developed and tested the NPS almost exclusively on evoked pain, nociceptive pain, also neuropathic and nociplastic? The second question is, this seems like it would be tremendously useful in drug development. Is it being used in that at all yet? And also for this differentiation diagnosis, functional pain syndromes or presumed functional pain syndromes.
Okay, thanks, Amanda. Yeah, the first question is about nociplastic pain and other kinds of applications to other cases and neuropathic pain. And I think we have this back pain study that we've been published a first paper on and are doing more. This is, I would consider nociplastic pain for lots of reasons. Tell you that. The short answer there is it doesn't track. It doesn't track, and we actually know from some other papers that the NPS tracks brief evoked pain. It doesn't track more sustained pain states, even if they're elicited by like sort of having, capsaicin in your mouth for a long period of time. So we think that there's actually a shift and something else is happening. There's other mediators that are coming into play when you go to longer term pain. So that's just the tip of the iceberg there, I think. We'd love to test. I think it's going to work well for, again, evoked, something where you have a lot of peripheral drive, and that could be neuropathic, and it could be, so we'd love to test that more. Drug discovery, I'll just say quickly, no, I've talked with the various different pharma companies. I think, we'll see. It's going to be sort of a pretty big commitment. And the thing is, this is good for hypersensitivity, allodynia. It's not necessarily that great for ongoing spontaneous chronic pain, where then we need more work. So we'll see.
Professor Glick.
Yeah, thanks for your talk. I'm curious if you think about this from a predictive coding perspective, like what are the brain regions and circuit networks coding for? And maybe you can just comment on that, like the idea of like, are they coding for the stimulus or are they coding for the difference between predicted and actual stimulus intensity? Or do you think about it work in that manner?
Yeah, so the question is about predictive coding and how does this map on and what are these circuits for? So I think a lot about it because I think that these ideas about predictive coding are very closely intertwined with the entire phenomenon of placebo effects. Although I sort of resisted talking about it in terms of predictive coding for a long time. I think one of the interesting angles here is that you can think of any placebo effect as a kind of example of predictive coding at some level. But then the question is, well, what's actually how far down do the prediction signals reach? What are they actually affecting, right? And I think our results actually speak to that. So one view is that the predictive model, that even at the spinal level, right, it's a prediction error that's being passed forward, and that's the predictive coding sort of idea. And I think that we can rule that out here. I think if that were true, we would see that NPS showing effects of all of these different kinds of manipulations. So I think there's multiple levels. So I think basically, yeah, I think that signal is really coding for the input more than anything else. It doesn't show strong prediction error related signals. It's not tracking prediction errors. This NPS, it's tracking pain related to the input. Anyway, so I'm sure we could talk more later. Yeah. Okay, okay. Is there any potential utilization of NPS in differential diagnosis? There are more than 30 different causes of low back pain, and their treatments are different. So accurate diagnosis is critical, right? And I would add to that. I would say that that typology is if it were based on a really super reliable pathophysiology, then we would be able to probably really effectively diagnose and treat back pain. But in some estimates that I've read, 80 to 90% of axial low back pain is idiopathic, and there isn't really a known structural cause. Now, people have a lot of opinions. Okay, so I'm sure somebody's going to hate me saying that. But the hope, yes, is that we can at least separate into this is the problem that's originating in nociceptive input in your back. That could be based on stenosis. It could be based on lupus propulsus, inflammation leaking out. It could be based on fascial, things or muscular things, but it's nociceptive. Versus the injury has healed, and what's left is what you could call a sort of memory of pain. Is the sensitization of the system, sensitization of avoidance systems. And we'd like to be able to separate those two things at least. I have to say one thing, and the proof is in the pudding. So I didn't talk about this today, but we did a treatment study for chronic low back pain as described, and a psychological and behavioral treatment worked beautifully for most of our people. We had people in chronic pain an average of 10 years, and after 4 weeks of behavioral treatment, they were 2/3 of them were pain-free or nearly pain-free, and they stayed that way for at least a year. So then it's like, okay, if the psychological behavioral treatment works that well, it's evidence that it wasn't, a nociceptive in your back cause, right? At least not only.
Wonderful. Thank you very much, Stuart.
Right.
With this, we
Thank you.